This is a session that includes a number of very accomplished speakers, and I will immediately start with the first one, who is a dear colleague of mine, namely Professor Roger Schibli from the Institute of Pharmaceutical Sciences at ETH Zurich. Roger is an inorganic chemist by training who received his PhD from the University of Basel in 1996. He then underwent postdoctoral training in radiochemistry in the United States and at the Paul Scherrer Institute in Villingen in Switzerland. In 2003, he then became an assistant professor for drug discovery technologies at ETH Zurich. At the same time, he was appointed a research group leader at the Paul Scherrer Institute. And after 6 years, in 2009, he was promoted to associate professor of radiopharmaceutical chemistry at ETH Zurich. And at the same time, he became the head of the Center of Radiopharmaceutical Sciences that is a shared operation by ETH, PSI, and the University Hospital. In 2019, then finally, he was promoted to full professor of radiopharmaceutical chemistry. Roger's research interests cover all aspects of radiopharmaceutical sciences, from diagnostic imaging to the development of radiotherapeutics, and he has a strong focus on the translation of research findings into applications. And I assume this is something that will be part of what he will be telling us today in his talk about an academic platform for translational radiotracer development for non-invasive imaging. Roger, the screen is for you to share.
Thank you very much, Karl-Heinz. I hope everybody can see my screen now.
I can.
Okay, perfect. So thank you very much for giving me the opportunity to talk about this project or this concept, which has been actually supported by the first round of PHT grants. And it's about a collaboration between ETH and PSI, specifically on the development of a translational platform for radiotracer development. So we are talking about applications or indications in nuclear medicine, because nuclear medicine actually, and I quote here a sentence from a recent publication in the European Journal of Nuclear Medicine, that nuclear medicine has actually the possibility to characterize biological processes at the cellular and the molecular level, and brings therefore information which maybe morphological imaging like MRI or a CT cannot provide. Therefore, it can actually make a significant contribution to personalized medicine in the future. And I hope if at the end of my talk, at least I give you a bit of a flavor where the journey could go. So we have indications in oncology, and typically neurology was a very early stage of nuclear medicine, was very much interested in tracer for non-invasive imaging of receptor density, receptor expression, but also in cardiology in the recent years, this has been also sparked a lot of attention. The reason why radiopharmaceuticals, radioactive material is so interesting for personalized medicine is because they have the technology has an extremely high sensitivity. So we can actually inject compounds at a very low concentration and still get nice pictures, as you can see here or here, for example. It is very specific, so we can actually infinitely develop radiotracers. So if we have a suitable target, we can identify a molecule which binds to this target. We can eventually radiolabel it with a radioactive isotope for diagnostic or even for therapeutic purposes. And therefore, it's high flexibility. So we do actually, we can actually explore the whole range of molecular space, so to say. However, radiopharmaceutical development is something which involves a lot of heavy infrastructure. So we need specific cyclotrons to produce those short-lived isotopes. We need, for translational research, so-called GMP facilities, so facilities to produce compounds according to good manufacturing practice. This, because of the short half-life, has to be very efficient. We have to obey, nevertheless, the law. We have to obey the Bundesamt für Gesundheit, the BAG, and also, of course, the cantonal, cantonale Heilmittelkontrolle to, being able to deliver such compounds for clinical application. On the other hand, also the nuclear physicians and the clinicians also have to operate and work with heavy infrastructures, scanners, combined MR, CT scanner with PET, also they have to obey, of course, the rules. And of course, if you want to do some clinical studies, you have also to, ask or permission by the clinical trial center, for example, here in Zurich. So this all leads to what I call the radiopharmaceutical value of death, because such platforms which allow the translation of preclinical traces into clinical setting is very rare. So we really have to, the interaction between the innovation of radiotracer development and whatever arrives at the clinical side is very rare these days. And this is mainly, particularly also in Switzerland, because we do not have sufficient expertise so far to do this efficiently. And that was actually the starting point why we would like to have a flexible platform for innovative radiopharmaceutical development. So the radiotracer development starts, as I mentioned, with cyclotrons. We need then to produce isotopes, separate those isotopes in specified and specific laboratories. Then we do the preclinical development, and a lot of groups around the world actually, they stay at this position where they have nice images in mice, but they never can translate that into clinics. And that's what we aim to, at this point, wanted to address by introducing a GMP platform. And I would say we had this before, we mentioned talk about that later on. We had already some good infrastructure in place, but we were not efficient to translate in a serious and in a good time frame things from mice to man. And so that's where we had the objectives, is actually that this technology transfer project 512, which was granted by the PHT initiative, should offer a support of different projects within the Swiss Personalized Health Initiative and the PHT project, and even beyond. So anybody who's interested in such radiotracers should have an access or need maybe new traces, and this platform should be able to produce these traces in a reasonable time frame, and also at the cost which is still affordable. So we wanted to implement the Swiss-wide concept of an automated radionuclide production, separation, incorporation according to good manufacturing practice for clinical research. The concept also was actually always aimed to involve clinical partners, academic researchers, and also industrial partners. And the output, what we wanted to, and what we would like to achieve, and have actually achieved, is that we have that we can conduct clinical research in Switzerland. We generate IP eventually by showing traces being actually suitable in, or do actually something in the clinics, so they are really suitable in clinical research, and also to spark also academic research, maybe leading back to the basic science. And that is the platform. The platform involved ETH Zurich as provider of the cyclotron, then also the GMP facilities. This is also at Paul Scherrer Institute, and we then installed, or actually wanted to do some improvement in terms of facility to automate this synthesis, and also to produce non-standard radionuclides. This is the projects which were actually engaged, or actually have been started, or have been started maybe slightly before this platform has been installed. So we had compounds or interest from the clinicians in Alzheimer traces, ALS, pain, systemic sclerosis, COVID, very recent COVID-19, hypoxia and depression, isotope production from the academic side and ovarian cancer, and also from the industrial side, there was interest in thyroid cancer therapy, and also pancreatic cancer monitoring, and many more. And that's where actually I'm going to show you some of those examples which we have now transferred, or via this platform have been able to transfer into clinical practice. So that's where the starting point. We actually at the ETH Zurich and also at PSI, we had already some heavy infrastructure, shielded hot cells in GMP quality. We had this automated but outdated systems. We also had a cyclotron at the ETH Zurich, which we actually can run 24/7 to produce isotopes, a situation which at the moment is not possible at the Paul Paul Scherrer Institute. So we had limited capacity to routine automation of isotope and radionuclide radiotracer production, as well as we were bound at the ETH with carbon-11 and fluor-18 radionuclides, but not, let's say, compounds or radionuclides like copper-67 and scandium-44, which could be of interest for different applications. The first step actually was to install heavy infrastructure. So we really had to kind of remodel some of the laboratories at the ETH. So we had to drill holes into solid concrete walls. We had to install this hot cell, an additional hot cell, and we also had to install the first solid target station in Switzerland to produce radionuclides, namely copper-64 and scandium-44. At the same time, we invest into new facilities to produce or automated systems to produce such radiotracers, and also new systems to fill such compounds under GMP conditions. First example was actually a nice example where the clinical partners were contacted a company which provided the tracer which allowed the imaging of the tau PET. This is namely this compound from Avid, which had been tested before in the clinics outside of Switzerland, but it was not available in Switzerland for clinical use. So they approached us, these three centers approached us, the platform, and say, well, would it be possible to adopt these tracers from the industry? Can you produce that for us to conduct a clinical study? And we could do actually that. This is an ongoing study still at the University Hospital in Geneva and also at CHUV, starting actually at the University Hospital of Zurich. You see here just a representative picture of a patient which has been imaged with this tracer, showing this accumulation of these tau tangles with this compound. Here, three-dimensional picture of such a patient. Up to now, we were able to produce more than 75 batches of this compound. More than 100 patients could be scanned. So we produced this compound at the ETH, transported it to the University Hospital in Geneva and also at CHUV, and two clinical studies, one supported by the Swiss National Science Foundation, which looked into the tau-related neurodegeneration, and also tau deposition in brain of elderly subjects, which is also a clinical study which is also being actually recorded in this, approved by the or let's say mentioned in the clinicaltrials.gov. So these studies are still ongoing. So we were very happy about this support, and also the clinicians are very happy about the reliability of this new platform. A second example is actually something which we developed at ETH, a preclinical development from starting in mice. And then actually we could actually convince three centers, University Hospital, CHUV, and Kantonsspital St. Gallen, and an additional center just joined recently, that they actually could test our compound in the clinics. This is a folic acid derivative, which is fluor-18 labeled here, and folic acid receptor overexpression in cancer is related to poor prognosis. And we performed this multi-center first-in-man study together with the clinicians, oncologists. Also, we did some correlation between immunohistochemistry, looking for folate receptor positivity or negativity, and we could clearly show that the immunohistochemistry in positive patients, that the compound is taken up in the majority of lesions, whereas the patients with negative immunohistochemistry, they did not take up the trace. So that was a study performed with 12 patients. It's a proof of concept study in lung cancer patients and ovarian cancer patients. And also we could do some dosimetry to show the safety of this compound. This has been recently been published in a European Journal of Nuclear Medicine Research. And due to this publication and also the early clinical studies and the results, it actually attracted attention from industry. So Merck is interested in licensing, or let's say sub-licensing, this compound together also with another industrial partner in Germany. So this is the next step, that this clinical first man study will be continues with a larger clinical study. Another study which was performed in this platform was together with the Paul Scherrer Institute, where and together with the University Hospital in Basel. This is now a therapeutic application. It's a mini gastrin analog targeting the CCK, so cholecystokinin receptor subtype 2, which is expressed on medullary thyroid cancer. Also, this was a proof of concept study together with this University Hospital of Basel, where we produced this compound at the Paul Scherrer Institute and transported this compound then to the hospital in Basel. And these are the first 6 patients where you can see clearly the uptake of the radiotracer in the lesions where this, the metastases are expressed or expressing this, this receptor, which can be addressed or can be labeled with this small peptide. This is has also been published recently, and also this compound, due to this first promising results in the clinic, actually attracted attention again from industry, and we were able to, or the Paul Scherrer Institute was able to recently license this compound to Debiopharm. Just want to like to highlight that this platform is therefore also very important actually to attract clinical or industrial partners, because nowadays if you come with a mouse study, usually it's very difficult to convince the industry to invest into such a larger endeavor. And this is also a compound which was at ETH developed, also here targeting a receptor of the glutamatergic system in the brain, namely subtype 5. This is the compound which has been developed at the ETH and together with the University Hospital and the University of Fribourg. We conducted a first in man and the following up the study actually, where you could see that this is the distribution, the heterogeneous distribution of the receptors in the brains of a healthy volunteers. Now this compound is being used in drug addict person, but also sleep deprivation studies were performed, where you can clearly see the receptor density, how it's changing over time. This could lead eventually to drug development, or to use this compound in the drug development of such, for example, addicted person. I would like to just mention also maybe a bit the expectations, what a clinician can expect when he would like to use this platform. And so I counted back, if you want to have the first patient injected, if everything is perfectly in place, so you have the radiosynthetic procedure and the analytical protocols are available, namely, for example, from an industrial partner, the tox data are available, clinical data, dosimetry are available, clinical protocols are written. You can expect within about 6 months that we could deliver maybe the first batch to your hospital. If, of course, if you're lacking some of the information, only toxicology data are available and the clinical dosimetry is available, maybe it's about 1 year you can expect. And if it's really a first in man where you only have data in mice, you have to actually, expect something like 18 months till you reach the first in man application. Also, when it comes to costs, if you at this stage, it costs you about 100,000, maybe from this point as 150,000, and you really start from scratch, you have to somehow expect the cost to 250,000, which is only radiopharmaceutical platform, how much it costs for the platform, without, of course, the clinical service which comes on top of that. This is quite, a bit of money, which of course is also one of the critical aspects I'm going to mention at the end of my talk. This is just a list of new projects which we are currently installing into the platform. It's 3 new tracers which we would like to produce. One is again a first in man study, targeting Alzheimer patients. We have here something which goes into a neuroinflammation with ALS patients. This is a tracer which is targeting the cannabinoid receptor subtype 2. And this is, again, it's an academic collaboration. It's a compound coming from the Stanford University, where the Cantonal Hospital in Basel is interested to use this compound for brain imaging, namely in endometriosis patients. So this is just the things which are currently ongoing, and also there's a collaboration, starting from this compound I mentioned before, the folic acid derivative, which now could be actually repurposed to an application in sclerotic patients, and also maybe also in COVID patients, because we have activated macrophages which are heavily involved in inflamed situations in the lungs, and this could be then also be used, this compound, which also targets the folate receptor beta on activating macrophages. This could also be used for this application. This is something which has submitted, has not yet been approved, but we are hoping that this, in the next couple of weeks, that we get a green light from the Swiss National Science Foundation. So I would at the end just briefly mention the challenges. Of course, the high costs. To maintain such clean rooms and to maintain certified equipment is extremely high. This has, is the reason we have to count or you have to expect to come up with a substantial amount of financial resources. Nevertheless, the production of such radiotracers, we feel they should be affordable for clinical partners in the future. So that is now the question, how to coming from this technology transfer project into a situation where the basics financing is sustainable, so the platform is still affordable for external users. And we're thinking that maybe this is something like a VC center could be a model why such a GMP platform could be maybe also implemented or maybe could be financed. Challenges are also the institutional collaborations, which, particularly translational aspects, are becoming more and more challenging. We're talking about personalized medicine, collaboration between the hospitals and ETH. Nevertheless, I've had the experience that these interactions are becoming actually even more difficult than actually easier in the near future. ETH domain is also somehow not prepared to do translational research, particularly if you collaborate with industry. They have other standards, and ETH, of course, is a research institution, so there is sometimes a clash between the two cultures, and we have actually a project which could not be performed because ETH could not fulfill or didn't want to fulfill the requirements from the industrial partner, although the clinical, there's a high interest in on the clinical side. And of course, the organization is challenging within the classical ETH structure. So as a professor, for example, it's very rare that you have to run such a platform by yourself. And it's also difficult to attract trained staff and keep them at the academic institution like ETH. And of course, the hurdles in GMP are not going down. So nevertheless, in conclusion, I think we could show that there is an interest of such a platform in Switzerland, that we could be able to build up capacity to provide free tracers in the past, to conduct clinical studies in Swiss hospitals. More than 200 patients were injected with tracers from this technology transfer project. The application of this solid target station, which I briefly mentioned in the beginning, is still due. We had some delay because of the Corona crisis, so the BAG was also not able to go all through this the paperwork and those doing on-site visits. We think that 2 additional radiotracers, or even 3, could be established by the end of this year. And for clinical studies, as I mentioned before, are being planned with the radiotracers developed on this platform. And I think also what is important, that clinical studies are more and more important to be selling arguments for licensing out new compounds. With that, I'm the end of the presentation. It's maybe a bit an unusual presentation in this series of talks, as I've seen. Nevertheless, I think it's important also that the technical aspects are important for personalized medicine. With that, I would like to thank all the people which I've mentioned here on this slide, and also particularly the Personalized Health and Related Technology funds, which were enabled to establish this platform. And I would like to thank you for your attention.